Okay, whenever you're ready.
Okay. Okay. Hello. Good morning, everybody. I'd like to have your attention and express my great pleasure to have with us today Professor Steven Klaine, who is visiting us. His real origin is Clemson University Institute of Environmental Toxicology, where he is a professor and interim director. But he came to us most immediately from Vietnam. Flew yesterday, into LAX and made time to come up over during his short stay in the LA area, made time to come to UCSB and to visit with us and to talk to us about his extensive work in nanotoxicology. So just a little bit about Steve. As I said, he's a professor and director at Clemson University Institute of Environmental Toxicology. His background is his PhD is in environmental science from Rice University, and his MS is also in environmental science from Rice. He did his bachelor's in biology at University of Cincinnati, and he has been a leader for over 30 years in studying and understanding the effects of contaminants in the environment. He has a very rich set of research contributions that he has made, including advancing the use of risk assessment in conjunction with his toxicological studies to put results in toxicology, ecotoxicology, into the perspective of making decisions. And that's something that we're very interested in within the Brin School, within the CEIN, within the context of environmental management in general. And I want to make mention of one important risk assessment that he performed is atrazine that was published a few years ago. It definitely was an important piece of work. And similarly, there's a review article that Klaine led. He's the first author on it. Many of us have read it. It's sort of a seminal review article in nano ecotoxicology. It's sort of the must-read article for anyone getting started in this work, which really speaks to his expertise, and that he is one of the original researchers in this area of nanotoxicology. With that, I just want to say thank you for making time for us. Our colleagues at UCLA and the UCEIN will be able to hear you today, and other collaborators in the CEIN. Steven is a part of the CEIN team. So his group and Steven are investigators with the Duke, Carnegie Mellon, CEIN. So we have sort of a sisterhood, brotherhood with you in that regard as well. Welcome. Thank you.
Thanks, Trish. And it really is no problem making time to come here. I was looking forward to visiting, and I've met some folks from here at meetings. And it turns out that the very first scientific meeting I went to as a graduate student was held here at the University of UC Santa Barbara. And I walked away thinking that there's no way you could do any work here because it's so beautiful. Obviously, you guys get a lot of work done, and but it still is very beautiful. So it's always a pleasure to come here to a nice part of the world and see some really interesting people. And I've had a great time discussing research with folks already this morning. So thanks again for the opportunity to come. And through the miracle of caffeine, I will try to not show the jet lag. Today, I'd like to talk about some of our nanomaterial research, and I'm going to give sort of a potpourri of 4 different research projects to try to illustrate some issues that I think are really important in ecotoxicology and nanomaterials. So having said that, with a little bit of introduction, I know nanomaterials shouldn't need any introduction to this audience, so I'm not going to have an extensive introduction. But I just want to point out with this slide that nanomaterials are used throughout society. So the issue is not is there exposure, because there is exposure. Folks are using nanomaterials. They're putting them on their skin. They're using them in products. They're getting into our ecosystem. And so, but the issue is, do we really know enough about the impacts? I point out that nanomaterials are important or unique for certain reasons. The first is their size, and you're all familiar with the size issue. But the other thing, and this second point here, is the issue of at this scale things become, different properties emerge that are not typical of that particular material at larger sizes. And so the whole issue of little pieces of carbon being stronger than steel, it probably wasn't even thought of decades ago, but now have emerged as of major use in society. The other issue that is going on in society is of importance to us as ecotoxicologists is that we're rapidly moving from lab scale production of these materials to larger scale. With the opening of the scaling of the CNano plant in China up to 500 tons per year of multi-wall carbon nanotubes, that's a significant amount of multi-wall nanotubes that are being produced. So use is going up, production is going up, and hence the potential for release is going up. Nanoscale control is being used in almost every sector of society. Here are just a few. I'm not going to go into them, but you can see that everywhere from tennis rackets to socks to iPod Nanos have nanomaterials in them and are present in our society. The real issue, though, is this gap in implications and applications. And I think that's what we're all about here, because as we discover new applications almost on a daily basis for these materials, it's our challenge to understand what the potential implications are. One might argue that, well, maybe we don't need to know that, but with the emergence of some data over the last few years, it's becoming obvious that while maybe all nanomaterials don't pose problems, and certainly we have our share of benign responses, there are folks that are demonstrating that there could be deleterious responses, as these folks from the UK showed in 2008 with macrophages and multi-wall nanotubes, showing a response similar to that has been shown with asbestos. So these particles get out into the environment, and they are well. This is sort of my sort of picture, or actually a picture I stole from Kathleen of production and human as well as ecological exposure. And exposure can occur in numerous ways out in the environment, either through deliberate application, through application of materials, say in landfills, where you have, or Superfund sites where you have organic contaminants, and they put zero-valent iron in to degrade those contaminants, or through inadvertent exposure through putting, say, titanium dioxide sunscreens on your skin and then jumping into the lake to go for a swim. So in any event, these materials move into the environment, and the consequence of this movement into the environment can be several different ways. Number one, humans can still be exposed, or the ecosystem can be exposed. And most of us that work in ecotoxicology are interested in the ecosystem, but ultimately we have to remember that humans are part of that ecosystem. So I have a couple different illustrations here of food chains. So just to remind you that not only can effects be direct on the ecosystem in terms of deleterious effect on a particular trophic level, but also these materials have the potential to be accumulated and move up the food chain through various levels of herbivores and carnivores, and then ultimately come back to human exposure again. So you can see these things are interconnected and sort of beg that they be identified or characterized, so that we don't really have unintended consequences. Because while everybody is really raving about the societal benefits of nanomaterials, it's important that we minimize what the problems are. So I want to move into some issues here that really underscore the rest of this talk. And I think the major issue here is that we're dealing with particles and not solutes. Toxicologists, I'd say over the last 50 years, have been used to dealing with solutes, whether you're dealing with drugs or whether you're dealing that's solubilized inside an organism, or whether you're dealing with environmental contaminants that solubilize in an aqueous environment. Most of the time we deal with solutes. And so dealing with particles has been a challenge, I would say, for ecotoxicologists. This is a picture of a pelagic invertebrate, Daphnia magna. Most of you are probably familiar with it. It's one of the white rats of aquatic toxicology. People use it a lot. We've used it for decades to look at the influence of solutes on this particular important organism, an important trophic level. We define bioavailability in many cases as the ability of the compound to move into the organism. And we define inhibitors of bioavailability as various water quality parameters that reduce that bioavailability. When you deal with particles, you're not dealing with solutes, you're dealing with particles. And these particles can aggregate, they can agglomerate, they can sediment, all which lead to varying exposure scenarios. So it's a challenge in many cases to quantify what the organism is actually exposed to. You put this organism with this, and you get things that look like this, which basically, as you can see here, in this case it's carbon nanotubes. These carbon nanotubes have not only aggregated, but adsorbed onto the surface of the Daphnia. This is certainly stressful to the organism. One can do this kind of research and actually generate things like LC50s. But there's no biochemical toxicity here, it's a physical stress. My point here is that unlike solutes, in which we actually look for sites and mechanisms of toxicity, in addition to doing that with particles, we need to characterize physical effects too. So things emerge that haven't really been on the forefront in terms of solute toxicity, and that is, particle characterization. All of a sudden, instead of collaborating with chemists, we find ourselves collaborating with physicists and material scientists. And that collaboration is critical to understanding exposure scenarios. Exposure characterization, exposure consistencies are both very critical if you're going to generate dose-response relationships. Remember that most of the toxicology that we do in the laboratory is comparative toxicology. We don't do it to predict what happens out in the environment because by definition, a small system is not very realistic, but it allows you to make comparisons between the toxicity of one material versus another, or the toxicity of the same material to two different organisms. So having this consistent exposure is very critical if you're going to generate numbers that others can use and repeat. And dose metric. All of a sudden, instead of solutes, I'm used to reporting solute toxicity as milligrams per liter, although my chemist friends always tell me molarity is better. So in cases where that's more appropriate, we use molarity. But bottom line is milligrams per liter the right dose metric? Or should we be looking at particle numbers per liter? Should we be looking at surface area per liter? So these are questions that are still not answered in this field. And finally, if you do toxicology work for a while, no matter what area of toxicology, you get used to using certain bioassays. You get used to using certain organisms. And when you're presented with a new problem, we oftentimes are guilty of going to the shelf, pulling the same bioassay we've used for years off the shelf, and using it to solve the new problem. So one could argue that maybe we're not even using the correct bioassays to study the effects of these particles. I'm going to try to address some of those issues in the next 40 minutes. So I'm going to talk about 4 different areas of research that we've been looking at. Some are a little bit more advanced than others, so bear with me if some of these areas are a little thin, but I think there's enough there to get the point across that I'm trying to make. Titanium dioxide, gold nanoparticles, and a couple different projects on carbon nanomaterials. The first one I'll talk about is this issue of titanium dioxide and are we using the correct bioassay system. This work really stemmed from some work that had been done by a visiting graduate student prior to him coming to my laboratory, and he had done some static bioassays with titanium dioxide and with Daphnia magna. And he had generated some dose response curves, and I took a look at them, and one of the things that struck me was how variable it was, and how variable he basically couldn't repeat the tests because things just weren't very reproducible in his system. And his static bioassay was the classic static bioassay. You take a beaker, you put in your exposure solution, and you then put in the Daphnia. You let it go for 48 hours, and you now have a 48-hour response. And you use the response metric, and he was using the initial concentration of the titanium dioxide. As many of you know who've worked with titanium dioxide, it's not very stable in suspension. And in fact, it sediments out pretty quickly. So we started thinking about what we could do to solve that, and maybe think about a different exposure scenario so that we could get more reproducible results. This work was done again with Daphnia magna, and this is his static system, okay? Here is a different system that we developed in order to test the toxicity of clay. It turns out if you go to the literature, and we've been doing some watershed work, and notice that eroding sediments are probably one of the biggest stressors in developing watersheds. If you go to the literature, there's not much data on the toxicity of suspended solids. I found it hard to believe, but there's a bit on salmon reproduction, but nothing on, very little on pelagic organisms. So we put together this system, and it's a fairly simple system that it's a 4-liter beaker that sits on a stirring plate, has a test tube rack that sits in the middle of it, I need to move this guy, but he seems to have migrated. Here's my Daphnia right here, and he should be in his glass tube. And she should be in his glass tube. And the Teflon mesh is at either end, it's sealed at the bottom, and then we hold it on with this glass ring at the top so that we can remove the glass ring, move the Teflon, and get to the organism. There's 10 of these that sit inside this rack. And by doing dye studies, you can determine if the system is completely mixed. The flow here actually moves up the sides of the container and down through the vessels in the middle. So the organisms are exposed and to a fairly constant concentration of the particulates. We've used this successfully for clay, and it's kind of hard to see in here because the lights are on, but there is a citation down here in January this year, Environmental Toxicology and Chemistry, where we report this using clay and some work we did with clay. We went ahead and set this up for using titanium dioxide. And the reason we did this in 2 ways. First was, we did this in 2 ways. We operated them all in a static way in which we just didn't have the stirring bars turned on, and then we did the same experiments with the stirring bars turned on. So we'd have, in the same system, we'd be able to mimic what happens either in a static system or in this stirred system. If you take samples within the water column and you just measure for titanium dioxide, you can see that the open circles and squares and triangles are the static system, and in this case, you can see that the percent remaining. Sorry about this being cut off. the percent remaining goes to practically zero by about 12 hours. So in a static system, all the work that's been done in the literature, and there's a significant amount of static bioassays in the literature that might report 48-hour numbers, but you know that the exposure is only gone for about 10 or 12 hours. The organisms are basically not exposed to titanium dioxide after that. Even in the third system, we've lost an appreciable amount, a measurable amount, but it was a bit more constant than the closed system. And of course, the amount you lose is really somewhat related to how much you have in there, and also related to particle size, which it's not too surprising. Now, toxicity data. The toxicity data here are the static numbers for the toxicity in terms of mortality, and you can see that they kind of jump around. And while these are pretty good confidence intervals for that particular run, and we had 3 replicates, if we went back and redid it, I assure you they wouldn't be identical. But one of the things that did come out is there was a pretty nice dose-response relationship in this third system. So we got a much more reproducible, and this, by the way, we did reproduce this, and this is much more reproducible, more consistent from test to test, and interestingly, if you go to the literature and look, this will be about the lowest LC50 number that you see for Daphnia magna and titanium dioxide. The reason, of course, is that they're actually being exposed to titanium dioxide for the entire 48 hours, and not for only a small part of it. So, the take-home message there is maybe the system isn't the right one to use for depending on what your question is, and also the need for this constant or this accurate characterization of exposure. If the exposure is always changing, then your dose response relationship is going to be suspect. And moreover, it's going to be difficult for people to repeat your work as well as compare your work to other work that's done. So the next thing I want to look at here or discuss is this work that we started doing with gold nanoparticles in aquatic food chains. Our overriding question here was, we started this work and we're just in the beginning parts of this, but our goal is to characterize uptake and distribution in an aquatic food chain of gold nanoparticles as a function of particle size, shape, and surface chemistry. And in this, what's fallen out pretty quickly is this issue of what's the correct exposure metric. And hopefully I'll be able to illustrate that. Of course, everyone's familiar with gold and the use of gold in ancient times. Here's a very famous piece of artwork. And this is, of course, the reflected light, the reflected color of the cup, and the transparent color of the cup. So, gold, colloidal gold has been used for a long time, and it's used in lots of different applications today in society. Also, just as an aside, it's not very toxic to aquatic organisms. We've had Daphnia magna swimming in grams per liter of suspended gold, and it seems to love it. Does the backstroke, has a lot of fun, doesn't seem to have a problem. So we thought it would be a nice organism to use in this case because we could differentiate uptake and distribution in a food chain from the toxic effects. So use 2 organisms. Solanacea capsuloidum is a unicellular green alga commonly used in aquatic bioassays, and it just so happens is the food that we feed the Daphnia magna on a daily basis. So we use 2 different types of gold nanoparticles, 18 nanometer and 4 nanometer spheres. I have a collaborator, Kathy Murphy at University of Illinois, who is a gold nanochemist. She produces very good nanoparticles, and it's important to get very reliable sources for nanomaterials. They are monodisperse. That is, when she sends us 18 nanometer gold spheres and we characterize them, they're all 18 nanometers. So it's very different than I've gotten other nanomaterials from suppliers which aren't like that. So water-only exposures, again with 2 organisms, and I'll show you the results with each of these separately. I'll also show you some depuration work with Daphnia magna, and foodborne uptake with solanestrum moving to the Daphnias. Sampled at various times during the exposures in order to look at rates of uptake, also rates of depuration, and organisms were sampled at each of those times for gold analysis to understand how much, or to characterize how much was in the organism. Same with the algae. They were grown in flasks, standard EPA medium, and samples taken in order to characterize how much how much gold was in the algae. These same algae were used as food in the foodborne study, in which case there was no aqueous exposure, just the exposure that came with the algae. In all cases, the organisms were harvested, dried, digested in aqua regia, and then analyzed on ICP-MS. We verified the sizes anyway of these materials. It's not only important to verify the particles that you get, but also in the solutions that you're using for exposures. So the sizes these were samples that were taken right from the algal medium or the Daphnia medium, and we used TEM to verify size and measured Zeta Potential in those same media, and minus 30 to minus 40, a fairly stable suspension, and so we had very little, did not see any visible sedimentation. So let's get to some results here. This is uptake of these nanoparticles with time by Daphnia magna from a waterborne exposure. And here I'm plotting micrograms per gram of, in the organism versus time. And you notice that this is 18 nanometer and this is 4 nanometer. So your conclusion from these data would be that 18 nanometer gold is taken up more efficiently than 4 nanometer gold. But does that really make sense? To me it didn't, and I got to thinking about it. And so if you replot this data, but you plot it as particle size, so because daphnids don't take up milligrams or micrograms of a substance, they take up particles. So if you then look at particle size, you see an entirely different picture. Particle size, the 4 nanometer is taken up very, very rapidly and very highly in terms of particle numbers, whereas on this scale it's kind of tough to see. There is a significant relationship here, and it is 18s are taken up, but at a much different rate on a particle basis. And of course, this is not rocket science. There's a whole lot more mass in an 18 nanometer particle than there is in a 4 nanometer particle. So, but putting them on a particle basis allows you to compare the results on a more level plane. Let's look at depuration. So here's the mass plot for depuration, showing the mass reduction for the 18s and very little change for the 4s. If you look at particle numbers, you see the same kind, although the 18, of course, is not as prominent as you'd see over here, but there is a reduction. But interestingly, there's no reduction in the amount of 4 nanometer gold particles that were in the organism, suggesting that while the 18 nanometer particles probably stayed within the gut tract of the daphnid and were simply excreted, so they were taken up and then excreted after the exposure was removed, then the 4s were taken up, but then simply were not excreted in the after removal. This suggests to us that the spores migrated out of the gut tract, and I was really hoping by now to have a picture to show that, and unfortunately I don't. So I apologize, this is rather new data, and I don't have a picture to verify where that gold is going, but it doesn't seem to be being excreted from the organism, at least not in this time frame. We are setting up a test to take these both exposures as well as depuration much longer. The algal uptake was kind of interesting. If you look at this data, it looks like it's all over the place. But what really seems to be happening here is that very rapid uptake, and even on this scale, it'd be rapid uptake, and then it levels off. And to me, this implies absorption and not movement into the organism. So what we've probably done here, even at 12 hours, is we've missed the critical part of this absorption isotherm, and we're going to go ahead and repeat this and get numbers down here. But the data suggests that it's taken up rapidly, adsorbed to the surface, and then it simply stays there. And the differential in numbers of particles is probably just simply a spatial issue with what is how much room there is on a surface of an algal cell. If you feed these algae to daphnids, you get what you'd expect. Uptake in a fairly consistent manner in terms of mass, probably because now there's as much mass on an algal cell of one versus the other. Since there's more 4 nanometer particles on that algal cell than there were 18, probably the mass is about the same on each algal cell, and that's why you see similar data for mass. Particles, you see the difference here, and that's just simply a measure of the different size, or a factor of a different size of the particles. So it looks like the size is important, okay, in these issues. At least the preliminary data suggests that, and that the 4 nanometer gold particles may not be effectively excreted, which suggests that they have some longer residence time in the organism, probably in the body cavity. But really what's important here is consider different metrics when you think about exposures and body burdens. And milligrams per mass may not be the appropriate measure for reporting your data for nanoparticles. We haven't looked at this data on a particle surface area basis, but I think that'd be worth exploring. The third project that I want to talk about is this whole issue of surface modified carbon nanomaterials. And I know that a number of you have been working with natural organic matter in some of your inorganic nanoparticles. And I want to talk about some of the work we've done with multi-wall carbon nanotubes and natural organic matter. Numerous carbon nanomaterials are out there, fullerenes, single-wall tubes, and multi-wall tubes. People have looked at their interaction with natural organic matter, at least multi-wall nanotubes, for quite a while. Sorry, I put this picture in here. They're used throughout society. Here you see numerous uses that you're probably used to knowing everywhere from materials and medicine to some stuff that maybe you're not used to seeing in terms of artificial muscles or desalination, and to things that people are just starting to envision here. In terms of their interactions with natural organic matter, what you see is here are several tubes that have different kinds of carbon nanomaterials and Swanee River natural organic matter. It's a common, standard source of natural organic matter. You can purchase it from the International Unique Substances Society, and because natural organic matter is so variable, it varies from site to site. Obviously, natural organic matter is that decomposition product of all the plants in the watershed, and so it's that key color that you get in surface waters. it's important to have at least a standard source so you can do your work. So this is the natural organic matter by itself, and this is with moving across from left to right, C60, C70, single wall tubes, multi-wall nanocoils, and nanowires. So you see pretty good stable suspensions with most of these materials. Of real note is this one with the single wall nanotubes. And not only did single wall nanotubes not stay suspended, they aggregated, precipitated, and took all the natural organic matter with it. So very interesting result. We haven't really followed up on that. We've really focused our work on multi-wall nanotubes and their interactions with organisms. Our premise here is that if a nanoparticle stays suspended in the water column, then the potential for exposure is going to be greater. And so the greater that exposure, the more likely it is to have some deleterious effect with a pelagic organism. Obviously, in cases where it doesn't stay in the water column, then the work that some of you are doing with biofilms would be very critical. And I think, again, a very critical part of ecosystems that need to be explored in terms of nanoparticle interactions. As I said, we used Swanee River natural organic matter for this work. We also isolated two of our own sources using the exact same technique that the International Human Substances Society uses. These are both, Swanee River is a coastal river in Georgia. These are both coastal rivers in South Carolina. So actually you'd expect the watersheds to be somewhat similar. We used 96-hour static renewal bioassays. In other words, we changed the exposure medium every 24 hours. So we didn't rely on long-term stability of these suspensions. We ensured that they were stable for at least 24 hours, and we simply made new suspensions every 24 hours. Again, kind of standard kinds of bioassay techniques here. We fed them just prior to renewal to minimize the time that food and nanoparticles were together in the system. So we wanted to minimize the effect of food on the effect of the nanomaterial. But because the bioassays went 96 hours, we had to feed them. The multi-wall nanotubes were produced by a colleague in the physics department, and here you see an electron micrograph of the as-produced material. Various dimensions here. We put those materials into an aqueous suspension of natural organic matter, sonicated them, allowed those suspensions to settle for 24 hours. So our experience with this is that you get absorption of the natural organic matter to the tube, and while it creates a stable suspension, it's not 100%. And so we wanted to account for anything that might sediment out during the bioassay. So before we used the suspensions, we allowed them to settle for 24 hours, and by characterizing the amount that settled, we were able to then get an accurate measure of how much was in the exposure suspension. We now have used this particular procedure for several years, and our colleagues, our collaborators have used this, and it turns out that it's a very reproducible procedure for producing a consistent exposure for these carbon wall or these carbon nanotubes. We characterize the suspensions in several different ways. Particle size and shape were characterized using dynamic light scattering as well as electron microscopy. Surface chemistry on the particles were characterized using Zeta Potential, and the Daphnias were characterized, as well as the materials themselves, were characterized using electron microscopy. This is a typical dose-response curve that we get in these particular bioassays. What I'm going to show you are the results of a whole lot of these. But here you have the control and in this case it's the Swanee River. control. You get no mortality here, and you get a concentration that produces no effects, and you get concentrations that do produce effects. With these data, you can produce LC50 numbers, and if you did that as a function of the amount of natural organic matter in the system that you use to suspend the particles with, you'd see that there's basically no significant difference. The average is about 2 to 2.5 milligrams per liter is the 96-hour LC50. And it doesn't matter whether you have less than 2 milligrams per liter of NOM or almost 20 milligrams per liter, suggesting to us that there's plenty of natural organic matter in natural waters to adsorb onto these materials and keep them at least moderately stable in suspension. We look at the different NOM sources. Here's the Black River. Here's the Swanee River. These were all nominal 15 milligrams per liter of natural organic matter. So you have these plotted by actual measured NOM concentrations, which varied a little bit. But notice that the Edison River was basically only half as toxic as the other two. So we then went on a needle in the haystack kind of search, looking for something to explain these data because, of course, as a skeptical major professor, I had the graduate students repeat this at least a dozen times. No, I was convinced after 3. But we looked at particle size. Particle size didn't change significantly with natural organic matter concentration, nor did it change among the different sources of natural organic matter. Zeta potential, excuse the fact that I've got fullerene data on this slide. I was lazy. I didn't want to separate it out. But the data you want to look at are the blue columns here, and you can see that there's no significant difference in the zeta potential, basically a measure of the stability of the suspension from the 3 different sources of natural organic matter. Obviously, when there's no natural organic matter, there's no stable suspension. These things are so hydrophobic that they aggregate and precipitate pretty quickly. I showed you a small picture of this. This is the as prepared. Before I get into this, I should also tell you that we used, we did IR spectra and C13 NMR analysis of the natural organic matter sources, both adsorbed and unabsorbed onto the particles. We still could not find a reason to explain why one particular source was less toxic than the other. We're still investigating that. These are the carbon nanotubes as prepared. If you sonicate them with natural organic matter, you get something that looks like this. In this case, we have, I think this is 15 milligrams per liter. And what you see here is what I suggested earlier, which was carbon nanotubes are very well coated with the natural organic matter, and you see excess natural organic matter sitting here because, of course, you have to draw the sample in order to get these pictures, and that causes the excess natural organic matter to kind of glob up. If you look a little closer, you see these things are in fact well coated. Tubes as well as tube bundles are well coated. We then look at the organism and the fate of these things within the gut tract of the organism. This is a controlled Daphnia magna, and basically sections across the gut tract. There's a fairly clean gut tract. This is the control, the microvilli and the lumen. Here's a little closer picture showing some algal cells, which basically the organism crushes and extracts the nutrients. That's its food source. If you then look at an exposed organism, here you see again the inside of the gut tract. In this case now it's full of nanotubes. Interestingly, the nanotubes kind of stay. They don't aggregate and become large clumps, but they do stay within the gut tract. They don't seem to get inside the microvilli region. on closer examination, here's a tube, and here are the microvilli. And again, they don't seem to. It doesn't seem to get into the tube or into the microvilli. We have not seen any movement of these materials out of the gut tract and into the organism. Although we've looked at lots of sections. Actually, I haven't looked. My students have looked. So, and it's quite tedious, but we have not been successful in finding movement out of the gut tract of these tubes. There's no doubt that these materials behave very differently when you have natural organic matter in the system. And there's not much data on non-modified carbon nanotubes, so it's kind of hard to compare our LC50 number with another number that are non-modified. But the number of 2 to 2.5 milligrams per liter is pretty low. We're rapidly coming, or not rapidly, but now coming to the conclusion that the nanotube toxicity in this case may simply be a function of energetics. In this case, carbon nanotubes clog the gut tract of the organism and simply prevent it from getting food. And of course, the thicker that clog is in the gut, the tougher it is for them to process food. So we started looking at gut clearance time. So basically defined as the amount of time it takes an organism after exposure, after they've been moved into clean water, the amount of time it takes for them to clear the gut tract. And you can see it visually under the microscope. As I told you earlier, we've looked at colloidal clay, and it had about 30 minutes on average. And we started to try to build up a number of these observations so we have a decent and average. It looks like for these multi-wall nanotubes, it's 28 hours, around 28 hours. So interestingly, the LC50, the 4-day LC50 for colloidal clay is around 100 milligrams per liter, and the 4-day LC50 for the multi-wall nanotube is about 2.5. So it's pretty easy to draw a straight line between two data points. So, but anyway, that's the direction I think is that we're headed in terms of looking at other kinds of nanoparticles and looking at not only toxicity but then how it relates to gut clearance time. And so I think a lot of these, again, particle effects may in fact be an energetic issue. And finally, no evidence that the carbon nanotubes cross the gut tract. The last bit of work, and just a couple more minutes that I want to talk about, is this issue of carbon nanoparticles serving as Trojan horses. What happens when these nanoparticles are in the solution with another contaminant? Do they make it more bioavailable? Do they enhance the toxicity? This is an issue that's been studied with, there's actually published work on carbon nanotubes and PAHs. We've done a lot of work in the past with ionic copper, and we know the toxicity of copper to Daphnia very well, so we decided to look at copper. In this case, it's very likely. We know that natural organic matter has lots of different ligands that for different metals. That's likely that maybe carbon nanotubes might also. So we, and of course, the nanotubes have to be modified in order to stay in suspension. So we thought that it's likely that there could be some interaction with the ionic copper. We looked at two different systems. The first system is single-wall nanotubes that were modified by a lysophospholipid. In this case, if you remember what phosphatidylcholine is, it's got a polar head group and two fatty acid tails. A lysophosphatidylcholine has only one of those tails. When you put that in with a single-wall nanotube, these tails line up along the carbon tube, and the heads stick out and form a fairly stable suspension, aqueous suspension. We looked at these stable single-wall nanotubes plus ionic copper and the toxicity to Daphnia magna. These are the dose response curves for the individual contaminants. So here you have copper by itself, 48-hour and 96-hour. And here you have the single-wall nanotubes by themselves, 48-hour, 96-hour, and 48-hour. And here you have the metrics up here, the LC50s. So these are easy to quantify individually. If you start looking at them together, there's numerous iterations of different kinds of combinations, which classically you do when you look at multiple contaminants, and you get lots of data. How do you make heads or tails out of this data? You simply, one way to do it, one approach, is to develop toxic units. So you simply divide through all the concentrations by the LC50s, which then normalizes the exposures for the copper as well as the single-wall nanotubes. You then plot them on as toxic units and observe versus expected. The line here is a one-to-one showing additivity, and you can see that basically all the data cluster around that line, suggesting that these two contaminants are in fact additive. So they're not. In this case, the nanoparticle is not increasing or decreasing the toxicity of the copper, or vice versa. However, if you measure the copper inside the organism, you'll find that at the two different copper concentrations here, that there's an increase, a significant, statistically significant increase in the amount of copper in each organism. What we believe is happening in this case is that the copper is being adsorbed to the lysophospholipid, and we now know that the Daphnia can strip off the lysophospholipid and use it as an energy source when it's exposed to these lysophospholipid-coated nanotubes. So in this case, while it didn't increase toxicity, it did serve as a different route of uptake for copper into the organism, and that was ingestion of the lysophospholipid with the copper. So you increase body burden. The second scenario in this case is the multi-wall nanotubes that had been surface modified by natural organic matter. In this case, we know that copper binds to various ligands in natural organic matter, and so would the additional multi-wall nanotubes change that and change the toxicity? Here are the data. It's kind of busy here, but let me walk you through this. This is at non-equilibrium, so this is right after the system is put together, the copper, the multi-wall nanotubes, and the natural organic matter. And look only at the clear bars, and you see that natural organic matter reduces the toxicity of the copper. Here's the copper concentration here and mortality. So you see that in many cases, particularly this one and this one, as well as the lowest one, the natural organic matter is by itself is significantly less than the others. When you add multi-wall nanotubes, you start to see the toxicity increase. Now, one of the issues here is this whole issue of equilibrium. We know that ionic copper and natural organic matter do not come to equilibrium in terms of ligand binding for at least 24 hours. So if you let this thing sit for 24 hours and then start the test again, or start the test at that point, you see entirely different data, and of course you see reduced mortality. And the reason you see reduced mortality is that now the copper has found the ligands, and the ligands have reduced the bioavailability. But one of the things that appears to be happening here, and you get the expected response with the NOM by itself. No toxicity, and then a measurable but small toxicity here. Now when you add the multi-wall nanotubes, what we believe is happening at this point, well, what we know is happening, is that the adsorption of the natural organic matter to the multi-wall nanotube is compromising the ligands that are serving to bind up the copper. So it's releasing the ionic copper. And we actually have ionic copper measurements that show this. And so now you've got higher amounts of ionic copper in these particular treatments, resulting in higher amounts of bioavailable copper and hence higher toxicity. So in this case, you don't have a Trojan horse in terms of carrying something into the organism, but the interaction with a water quality component that normally reduces bioavailability actually ends up increasing the bioavailability. Why is this important? Well, I don't know if you're familiar with a model called the biotic ligand model, but the biotic ligand model is a model designed to predict the bioavailability of copper and other metals, but copper is important in this case, to aquatic organisms. And in fact, the US EPA uses the biotic ligand model to determine site-specific water quality criteria for copper. So the addition of a little bit of nanotubes is going to compromise the ability of that biotic ligand model to accurately predict the bioavailable fraction of copper. So that really concludes a lot of the messages that I wanted to get across. I'd like to acknowledge my postdoc, Aaron Roberts, who now is at the University of North Texas, visiting graduate student Kathy Kim from GIST in Korea. My students now do all the work, numerous graduate students, my colleagues. These guys, my students do all the work, they simply let me talk about it. And funding from the US EPA, a couple of different STAR grants, from Clemson University, the public service activities, as well as Office of Research, and of course, SAIN for inspiration. And here's my group, my lab group, current lab group. I'm the old guy in the back, and like to invite you all to consider coming to Nano 2010. It's the first time this particular conference will be held in the U.S., and it will, in fact, be the last time for at least 5 years because this thing moves around the world. So it's primarily been in UK and in Europe, but we have designs to take it to Australia and Asia in the next few years. So this will be the only time it'll be in the U.S., well, probably for at least 4 or 5 more years. So I would love to see you in Clemson, and I guarantee you it'll be a good time. So with that, I'd like to take any questions you may have. So. Yes.
Before we go back to your presentation about taking back like Daphnia, and you were discussing the importance of surface association of the nanoparticle with the Daphnia. And I just wanted to understand, is your perspective that maybe in particular with TiO2, that surface association is a necessary preceding phenomenon before, in order for...
The moderator has disconnected. The conference will now end.